Hello, everyone. Thank you for joining us today for this webinar. My name is Griffey. I am the Chief Operating Officer for StemBioSys, located here in San Antonio, Texas. What I'm going to speak to you about today, I'm going to be describing a technology that we've developed at StemBioSys that we feel is the next evolution in stem cell research, and specifically stem cell culture. Our technology addresses the importance of the cell extracellular matrix interaction. I'd like to start with this slide. This was kindly provided to me by Dr. Robert Meacham at Washington University in St. Louis. This is a freeze fracture image, and you can see in the top right the extracellular matrix, and in the bottom left a cell. And specifically, I like to point people to that junction. It goes from top left to bottom right, showing all of the interaction between the extracellular matrix and a cell. That's a lot of information, and it's not just to hold the cell in place. There's a significant information exchange between the extracellular matrix and the cell, and that's what I'm speaking primarily to about this technology today. So we have known for some time, and it's been published widely in the scientific literature about not only just the importance of cell extracellular matrix interaction, but also specific to stem cells. And we've just thrown a few examples up here. While we've known about this and accept that key role that the extracellular matrix plays in directing cell behavior, we continue to remove cells from this native environment when we want to place them in culture. So essentially, we're ripping these cells out of their normal environment in the body and putting them into cell culture. In most cases, we put them onto a foreign substance or a foreign body, which is polystyrene or plastic. Sometimes we use some proteins, and I'll mention that as well. But expecting these cells to behave normally once we've done that, I think is a bit naive, and that's also what this technology addresses. So let's look at the methods for cell culture and how they've evolved over the years. Looking at this slide on the bottom left, in the 40s, we were using jars a lot of times to do cell culture. Going forward to the 50s, glass petri dishes came in, and a lot of times these glass dishes were coated with different substrates, but that was the primary method of use at the time. And then about 10 years later, polystyrene really changed the way we did cell culture because it was very easy to produce, a very standard substrate. But that really hasn't changed much over the past 40 to 50 years. And polystyrene, as I mentioned, is a very different substrate. It's a synthetic that cells don't normally come in contact with. So what we've developed is a natural environment created by stem cells primarily for stem cell growth. So what I'll show here, and I'll stop speaking, it's got its own audio. This is a video, kind of a cartoon that will describe how we make our bone marrow high-performance microenvironment.
StemBioSys creates on a culture dish a stem cell niche similar to that found in healthy human bone marrow. The following illustrates the process for creating our high-performance microenvironment, or HPME. We begin with a bank of commercially available bone marrow stromal cells from healthy young adults. These cells are then added to a standard cell culture dish. We add a proprietary culture medium, and the cells begin to divide, increasing cell numbers and fully covering the surface of the dish or other culture substrate. The cells are then stimulated to produce an extracellular matrix. These matrix proteins are deposited in a defined and ordered three-dimensional structure, a home built by the bone marrow cells in a fashion similar to what they do in their native bone marrow. This extracellular matrix is a complex, multi-protein composite that mimics the natural bone marrow niche for stem cells. Following production of their extracellular matrix, the remaining bone marrow stromal cells are removed very gently so as not to change the architecture or the biochemistry produced by the cells. The matrix is then thoroughly washed, the remaining solutions are removed, and the dishes or vessels dried, leaving on the surface of the cell culture substrate a native, three-dimensional home for stem cell proliferation similar to that from which they came.
This is not an easy approach, as you can tell, but we feel it's by far the most natural approach for culturing stem cells. So we take that extra care and take these extra steps to produce this high-performance microenvironment. Analysis of this finished product confirms the complex biologic structure and biochemistry of a cell-produced extracellular matrix. There's well over 100 different proteins present, which is being laid down by cells in a defined and orchestrated manner. So this is an extracellular matrix produced by stem cells for stem cells. Being a complex extracellular matrix, there's also concern about the ability to reproduce the same extracellular matrix from batch to batch. And I think the slide illustrated here is a great example of how mesenchymal stem cell behavior has been normalized by providing this extracellular matrix. And this is compared to a very standard substrate. In this case, it's tissue culture treated plastic. On the x-axis of this graph are different donors, bone marrow cell donors. And on the y-axis is the doubling time. So what you see, not only are the cells grown on bone marrow HPME dividing much more rapidly, it's a much more consistent behavior that you see from these cells. As opposed to those placed on plastic, the proliferation rate varies from donor to donor, and that's probably indicative of them interacting with what is a foreign substance, that being tissue culture treated plastic. Further evidence of the powerful effects that extracellular matrix has on cell behavior is depicted in this gene map, which illustrates the differences in the gene expression profiles of the same cells grown either on plastic, depicted on the right-hand side, versus our bone marrow high-performance microenvironment. Looking at the functional characteristics of these MSCs, you can see that there is a marked difference of these cells when they're interacting with the different substrates. Functionally, we see much better cell attachment and stem cell isolation. The example given here is using human cord blood mononuclear cells that have been plated either on tissue culture treated plastic or on our bone marrow high performance microenvironment. You can see a lot more cells attached. That leads to a better isolation of the mesenchymal stem cells, which we were after in this case. When compared to other substrates, MSCs grown on HPME have an increased cell proliferation rate, smaller cell size, which is index of stem cell potency, and a higher percentage of these cells are expressing SSCA4 through more population doublings when compared to some of these other substrates. The comparisons that we have here are tissue culture treated plastic on the left, our HPME, fibronectin, a product from Cell Start, and the other product is Mesencult. And you'll notice on the far right-hand graph, the SSCA4 expression, we're not showing a result for cells grown on Mesencult. And that was only because we were not able to isolate and grow enough cells using that product to do that final analysis. So the data that I've spoken to previously has been primarily looking at bone marrow mesenchymal stem cells. But the effects that we see with our bone marrow high performance microenvironment are not limited only to bone marrow cells. The examples here are cells, mesenchymal stem cells isolated from cord blood and from adipose tissue. But we've also taken mesenchymal stem cells from cord tissue and perinatal tissue sources. And we're seeing the same results. We're seeing increased proliferation, small cell size, and higher expression of SSCA4. These are some examples of the other extracellular matrix protein products that are available for cell culture applications. BioCoat, which provides several different types of substrates, generally with one extracellular matrix protein, which has been pre-coated onto these slides to culture cells. Cell Start and Mesencult were products developed specifically for mesenchymal stem cell growth. And then, of course, Matrigel, which is derived from a mouse tumor cell line. These are all soluble extracellular matrix proteins. So the protein has been extracted and purified so that they can pull out certain one-off extracellular matrix proteins. As compared to our bone marrow high-performance microenvironment, which we mentioned, there's well over 100 different proteins. We think, obviously, this is a much different substrate and much more natural than the others that are currently available. The advantages of our bone marrow high-performance microenvironment for mesenchymal stem cell culture versus other products is outlined here. The product is ready to use after just a brief rehydration step, and it leads to faster proliferation and better maintenance of mesenchymal stem cell phenotype. Despite these advantages, we've been very careful to price this product similar to those that are also listed on this sheet. Illustrated here on this slide is an example of optimized cell isolation and growth of primary cells using some of these competitive products. In this example, we've followed the various manufacturers' recommendations for media and attachment factor in doing the comparison. This is to tissue culture plastic. Our standard HPME, a new product which we're developing and should have out at the beginning of 2018, is a xeno-free version of our high-performance microenvironment. The BioCoat products, specifically fibronectin, CellStart, and Mesencult. So in summary, artificial substrates show a disregard for the current understanding of stem cell interactions. Cells change in response to signals from simple matrices. There is no surface which is signal-free, including cell culture-treated plastic. Mesenchymal stem cells grown on our bone marrow high-performance microenvironment are more proliferative and more active than cells on other available substrates. Mesenchymal stem cells grown on our BM HPME appear to be more responsive to growth factors as well. And finally, mesenchymal stem cells grown on the bone marrow high-performance microenvironment exhibit enhanced stem cell potency characteristics. So finally, I'd also like to just mention that we're about to release a couple of different cell lines which we've been able to isolate using this HPME technology. These cells appear to be relatively rare, and we find that they grow much better, and we're able to isolate these cells using our HPME technology. One of these cell lines will be an endothelial progenitor cell isolated from human umbilical cord blood, and the second cell line, available later this year, comes from a perinatal tissue source. So finally, I'd like to thank you again for viewing our webinar today, and I hope you have a great day. Thank you.